students, welcome to the PD Life Science Academy for the Happy Learning. NEET एक्झामचे प्रिपरेशन जे विद्यार्थी करत आहेत त्यांच्यासाठी बेसिक कन्सेप्ट लर्निंग आणि लिंकिंग ऑफ कन्सेप्ट हे जेवढे गरजेचे आहे तेवढेच NCERT बुकमधील चॅप्टरचे कंटेंट रीड करणे आवश्यक आहे. हे करत असताना फक्त एक रीडिंग करून चालणार नाही, मल्टिपल टाइम्स रिव्हिजन होणे गरजेचे आहे. यात टाळाटाळ करणे चांगले नव्हे. आपण सेट केलेले टार्गेट अचीव करण्यासाठी PD लाईफ सायन्स अकॅडमीने सर्व स्टुडंटसाठी जे नीट एक्झामचे प्रिपरेशन करीत आहेत त्यांच्यासाठी 21 डे रीडिंग चॅलेंजची सुरुवात केली आहे. रोज एका चॅप्टरचे रीडिंग आपण कंप्लीट करणार आहोत. हे चॅलेंज ज्या विद्यार्थ्यांनी ॲक्सेप्ट केले आहे त्यांनी त्यांचे डेली टार्गेट अचीव केल्यावर कॉमेंट करून सांगायचे आहे. सो लेट्स गेट रेडी फॉर टेकिंग रीडिंग चॅलेंज. ऑल द व्हेरी बेस्ट. थँक यू.
21 डे biology reading challenge, day 12 reading challenge, chapter number 12 from NCERT biology textbook of 11th standard. Chapter number 11, name of chapter is a mineral nutrition. The basic need of all living organisms are essentially the same. They require the macromolecules such as the carbohydrate, proteins and fats, and the water and the minerals for their growth and the development. This chapter focuses mainly on the inorganic plant nutrients, wherein you will study the methods of the identifying the element essential to growth and the development of the plant and the criteria for establishing the essentially. You will also study the role of the essential element, their major deficiency symptoms and the mechanism of the absorption of their essential elements. The chapter also introduces you briefly to the significance and the mechanism of the biological nitrogen fixation. Now the 12.1 is a method to study the mineral requirement of the plant. In 1860, the Julius van Sach, a prominent German botanist, demonstrated for the first time the plant could be grown in a maturity in the definite nutrient solution in a complete absence of the soil, and this technique is a growing the plant in the nutrient solution is known as a hydroponics. Since then, the number of improved methods have been employed to try and determine the mineral nutrient essential for the plant. Essence of all these methods involves the culture of the plant in a soil free then defined mineral solution, and these methods requires a purified water and the mineral nutrient salts. Can you explain why is this so essential? After a series of experiments in which the roots of the plant were immersed in the nutrient solution and wherein elements are added or a substitute or removed or given in a varied concentration, a mineral solution suitable for the plant growth as obtained. By this method, essential elements were identified and their deficiency symptoms discovered. The hydroponic has been successfully embedded as a technique for the commercial production of the vegetable such as a tomato, seedless cucumber or a lettuce. It must be emphasized that the nutrient solution must be adequately aerated to obtain an optimum growth. What would happen if the solution were the poorly aerated? Diagrammatic view of the hydroponics technique is given in the figure. Now the 12.2 is an essential mineral element. Most of the mineral present in the soil can either plants through the roots. In fact, the more than the 60 elements of the 105 discovered as far as found in the different plants. Some plant species accumulate a selenium, some other are the gold, while some plants growing near a nuclear test site take up a radioactive strontium. There are the techniques that are able to detect the minerals even at a very low concentration, that is the 10 raised to minus 8 grams per ml. The question is whether all the diverse mineral element present in plant, for example the gold and the selenium are the mentioned above, are they really necessary for the plant? How do we decide what is essential for the plant and what is not? Now the 12.2.1 is a criteria for essentiality. The criteria for essentiality of an element are given below. The first is the element must be absolutely necessary for supporting a normal growth and the reproduction. In absence of the element, the plant do not complete their life cycle or set a seed. Now the second is the requirement of the element must be specific and not replaceable by another element. In other words, deficiency of any one element cannot be met by the supplying some other element. The element must be directly involved in the metabolism of the plant. This is the third criteria. Then based upon the above criteria, only a few elements have been found to be absolutely essential for the plant growth and the metabolism. And these elements are further divided into the two broad categories based on their quantitative requirement. That is, first is a macro element and second is a micro element. Now the macro elements are generally present in the plant tissues in a large amount, that is in excess of the 10 millimolar per kg of the dry matter. The macronutrients include the calcium, carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur, potassium, calcium and the magnesium. Of these, the carbon, hydrogen and the oxygen are mainly obtained from the CO2 and the water, while the other are absorbed from the soil as a mineral nutrients. Then the second are the micronutrients, or it is also known as a trace element, are needed in a very small amount, that is less than the 10 millimolar per kg of the dry weight. This include the iron, manganese, copper, molybdenum, zinc, boron, chlorine and the nickel. In addition to the 17 essential elements named above, there are some beneficial elements such as the sodium, silicon, cobalt and the selenium. They are required by the higher plants. Essential elements can also be grouped into the four broad categories on the basis of their derived functions, and these categories are: first is the essential element as a component of the biomolecule and hence the structural element of the cell, for example carbon, hydrogen, oxygen and the nitrogen. Now the second is the essential element that is a component of the energy related chemical compound in a plant, for example the magnesium in the chlorophyll and the phosphorus in a ATP. Now third is the essential element that activate or inhibit the enzyme. For example Mg2+ is an activator of both the ribulose bisphosphate carboxylase oxygenase and the phosphoenol pyruvate carboxylase, both of which are the critical enzyme in a photosynthetic carbon fixation. Now the zinc is the activator of the alcohol dehydrogenase or a molybdenum of the nitrogenase during the nitrogen metabolism. Can you name a few more elements that falls in this category? For this, you will need to recollect the sum of the biochemical pathways you have studied earlier. Now fourth is some essential element can alter the osmotic potential of the cell. The potassium plays an important role in the opening and the closing of the stomata, and you may recall the role of the minerals and the solute in determining the water potential of the cell. Now the 12.2.2 is a role of the macro and the micro element, macro and the micronutrients. The essential element perform a several function and they participate in the various metabolic processes in the plant cell, such as the permeability of the cell membrane, maintenance of the osmotic concentration of the cell sap, electron transport system, buffering action, enzymatic activity, and act as a major constituents of the macromolecule and the coenzyme. Various forms and the functions of the essential nutrient element are given below. The first is a nitrogen. This is the essential nutrient element required by the plant in a greatest amount. It is absorbed mainly as a NO3 minus. Through the some are also taken up, that is the NO2 minus or NH4 plus. The nitrogen is required by all parts of the plant, particularly the meristematic tissue and the metabolically active cell. The nitrogen is one of the major constituents of the protein, nucleic acid, vitamins and the hormones. Now second is a phosphorus. Phosphorus is absorbed by the plant from the soil in the form of the phosphate ion, either as a H2PO4 minus or HPO4 minus 2. The phosphorus is a constituent of the cell membrane, certain proteins, all nucleic acid and the nucleotides, and is required for all phosphorylation reaction. Now the potassium. It is absorbed as a potassium ion in a plant. This is required in a more abundant quantity in the meristematic tissue, buds, leaves and the root tip. The potassium helps to maintain the anion exchange balancing in the cell and is involved in the protein synthesis, opening and the closing of the stomata, activation of the enzymes and in the maintenance of the turgidity of the cell. Now next is the calcium. The plant absorbs the calcium from the soil in the form of the calcium ion, that is Ca2 plus. The calcium is required by the meristematic and the differentiated tissues. During the cell division, it is used in the synthesis of the cell wall, particularly as a calcium pectate in the middle lamella. It is also needed during the formation of the mitotic spindle fibers. It accumulates in older leaves and it involves in the normal functioning of the cell membrane. It activates a certain enzyme and plays an important role in the regulating the metabolic activities. Now next one is the magnesium. It is absorbed by the plant in the form of divalent, that is the Mg2 plus. It activates the enzyme of the respiration, photosynthesis and are involved in the synthesis of DNA and the RNA. Magnesium is a constituent of the ring structure of the chlorophyll and helps to maintain the ribosome structure. Now the sulfur. The plant obtains the sulfur in the form of the sulfate, that is SO4 2 minus. The sulfur is present in the two amino acid, that is the cysteine and the methionine, and is a main constituent of the several coenzyme vitamins, that is the thiamine, biotin, coenzyme A and ferredoxin. Now the iron. Plant obtain the iron in the form of the ferric ion, that is the Fe3 plus. It is required in the larger amount in the comparison of other micronutrients. It is an important constituent of the protein involved in the transfer of the electron, like ferredoxin, cytochrome and the cytochrome. It reveals the oxidized form from the Fe2 plus to the Fe3 plus during the electron transfer. It activates the catalase enzyme and is essential for the formation of the chlorophyll pigment. Now the manganese. It is absorbed in the form of the manganese ions, that is the Mn2 plus, and it activates the many enzymes involved in the photosynthesis, respiration and nitrogen metabolism. The best defined function of the manganese is in the splitting of the water to liberate the oxygen during the photosynthesis. Now the zinc. The plant obtain a zinc as a Zn2 plus ion. It activates various enzymes, especially the carboxylase. It also need in the synthesis of the oxygen. Now the copper. It is absorbed as a cupric ion, that is the Cu2 plus. It is essential for the overall metabolism in the plant. Like ions, it is associated with a certain enzymes involved in the redox reaction and is reversible, oxidized from Cu2 plus Cu plus to Cu2 plus. Now the boron. It is absorbed by the BO3 3 minus or B4 O7 2-. The boron is required for the uptake and the utilization of the calcium, that is the Ca2+, the membrane functioning, the pollen germination, cell elongation, cell differentiation, and the carbohydrate translocation. Now the molybdenum, the plant obtain it in the form of the molybdenum ions, that is the MoO2 2+. It is a component of the several enzymes including the nitrogenase, nitrate, reductase, both of which are participated in the nitrogen metabolism. Now last one is a chlorine. It is absorbed in the form of the chloride ion, that is the Cl-. Along with the sodium and the potassium, it helps in the determining the solute concentration and the anion cation balance in the cell. It is essential for the water splitting reaction in the photosynthesis, the reaction that leads to the oxygen evolved. Now the 12.2.3 is a deficiency symptom of the essential elements. Whether the supply of the essential element become the limited, the plant growth get retarded. The concentration of the essential element below which the plant growth is retarded, the term as a critical concentration. The element is said to be the deficiency when the present below the critical concentration. Since each element has the one or a more specific structural and the functional role in a plant. In absence of any particular element, the plant shows a certain morphological changes. These morphological changes are indicative of the certain element deficiencies and are called as a deficiency symptoms. The deficiency symptoms vary from the element to element and they disappear when the deficiency mineral nutrients is provided to the plant. However, if the deprivation continue, it may eventually lead to the death of the plant. The parts of the plant that shows the deficiency symptom also depends on the mobility of the element in the plant. For elements that are actively mobilized within the plant and exported to the young developing tissues, the deficiency symptom tends to appear first in older tissues. For example, the deficiency symptom of the nitrogen, potassium, magnesium, and the visible first in the senescence leaves. In the older leaves, the biomolecule containing these elements are broken down, making these elements available for the mobilizing to the younger leaves. The deficiency symptom tends to appear first in the young tissues whenever the elements are relatively immobile and are not transported out of the mature organ. For example, the element like the sulfur and the calcium are the part of the structural component of the cell and hence are not easily released. And this aspect of the mineral nutrient of the plant is of the great significance and the importance of the agriculture and the horticulture. The kind of the deficiency symptoms shown in the plant include the chlorosis, necrosis, stunted plant growth, premature fall of the leaves and the buds, and the imbibition of the cell division. Chlorosis is the loss of the chlorophyll leading to the yellowing of the leaves, and this symptom is caused by the deficiency element, that is the sodium, that is the nitrogen, then potassium, magnesium, sulfur, ferrous, then manganese, zinc, and molybdenum. Likewise, the necrosis or the death of the tissue, particularly the leaf tissue, due to the deficiency of the calcium, magnesium, copper, and the potassium. Lack of the low level of the nitrogen, potassium, sulfur, and molybdenum causes the inhibition of the cell division, and some element like the nitrogen and sulfur, molybdenum delay the flowering of their concentration in the plant is low. You can see from the above the deficiency of the any element can causes the multiple symptoms, and that the same symptom may be caused by the deficiency of the one of the several different element. Hence, to identify the deficiency element, one has to study all the symptoms developed in all the various parts of the plant and compare them with the available standard table. We must also be aware the different plants also responsible for the differently to the deficiency of the same element. 12.2.4 is a toxicity of the micronutrient. The requirement of the micronutrient is always in a low amount, while their moderate decrease causes the deficiency symptom and the moderate increase causes the toxicity. In other words, there is a narrow range of the concentration at which the elements are optimum. Any mineral ion concentration in the tissue that reduces the dry weight of the tissue by about the 10% is considered the toxic. Such a critical concentration varied widely along the different micronutrients. The toxicity symptoms are the difficult to identify. The toxicity level for any element also vary for the different plants. Many times the excess of an element may inhibit the uptake of the another element. For example, the prominent symptom of the manganese toxicity is the appearance of the brown spot surrounded by the chlorotic veins. It is important to know that the manganese compete with the iron and magnesium for uptake, and with the magnesium for the binding with the enzyme. The manganese also inhibit the calcium translocation in the shoot apex. Therefore, the excess of the manganese may in fact induce the deficiency of the iron, the magnesium and the calcium. Thus, what appear as a symptom of the manganese toxicity may actually be a deficiency symptom of the iron, magnesium and the calcium. Can this knowledge be the some important to the farmer, the gardener, or even for you in your kitchen garden? Now the 12.3 is a mechanism of the absorption of element. Much of the studies on the mechanism of the absorption of the element by the plant has been carried out in isolated cells, tissues or organ. These studies reveal that the process of the absorption can be demarcated into the two main phases. In the first phase, the initial rapid uptake of the ions into the free space or the outer space of the cell, the apoplast, is a passive. In a second phase, the uptake of the ion take in a slow into a inner space, that is the symplast of the cell. The passive movement of the ion into the apoplast usually occurs through the ion channel, the transport membrane protein that function as a selective pores. On the other hand, the entry or exit ions of and from the symplast requires the expenditure of the metabolic energy, which is an active process. The movement of the ion is usually called as a flux. The inward movement into the cell is a influx, the outward movement. The influx, you have read an aspect of the mineral nutrient uptake and the translocation in the plant in chapter number 11. Now the 12.4 is a translocation of the solids. The mineral salts are translocated through the xylem along with the ascending streams of the water, which is pulled up through the plant by the transpiration pool. Analysis of the xylem sap shows the presence of the mineral salt in it. Use the radioisotope and the mineral element also substantiate the view that they are transported through the xylem. You have already discussed the movement of the water in the xylem in chapter number 11. Now the 12.5 is a soil as a reservoir of the essential element. Majority of the nutrients that are essential for the growth and the development of the plant become available to the root due to the weathering and the breaking of the rocks. These processes enrich the soil with the dissolving the ions and inorganic salts. Since they are derived from the rock minerals, their role of the plant nutrition is refers to as the mineral nutrition. The soil consists of the wide variety of the substances. The soil not only supplies the mineral but also harbors the nitrogen fixing bacteria, other microbes, holds the water and supplies the air to the root and act as a matrix that stabilizes the plant. Since the deficiency of the essential mineral affects the crop yield and there is often a need for the supplying them through the fertilizer. both the macronutrients and the macronutrients like the NPK and sulfur, etc., and the micronutrient like the copper, then the zinc, ferrous, manganese, etc., forms the compound of the fertilizer and applies as per need. Now the 12.6 is the metabolism of the nitrogen. Now the 12.6.1 is a nitrogen cycle. Apart from the carbon, hydrogen and oxygen, the nitrogen is the most prevalent element in the living organism. Nitrogen is a constituent of the amino acid, protein, hormones, chlorophyll and the many of the vitamins. The plant competes with the microbes for the limiting nitrogen that available in the soil. This nitrogen is a limiting nutrient for both the natural and the agriculture ecosystem. Nitrogen exists as a two nitrogen atom joined by the very strong triple covalent bond, that is the N triple bond N. The process of conversion of the nitrogen gas, that is the N2, to the ammonia is a term as a nitrogen fixation. In nature, the lightning and the ultraviolet radiation provides the enough energy to convert the nitrogen to the nitrogen oxide, that is the NO, NO2, N2O, etc. The industrial combustion or forest fires, automobile exhaust and power generating stations are also source of the atmospheric nitrogen oxides. The decomposition of the organic nitrogen of the dead plant and the animals into the ammonia is called as ammonification. Some of the ammonia volatilizes and re-enters the atmosphere, but most of it converted into the nitrate by the soil bacteria as the following steps. That is the 2NH3 plus 3O2, it gives rise to the 2NO2 minus plus 2H plus 2H2O. Now 2NO2 minus plus O2, it gives rise to the 2NO3 minus. Ammonia is first oxidized to the nitrate by the bacteria Nitrosomonas and or the Nitrosococcus. The nitrite is further oxidized to the nitrite with the help of the bacterium, that is the Nitrobacter. These steps are called as a nitrification. These nitrifying bacteria are the chemoautotroph. The nitrate thus formed is absorbed by the plant and transported to the leaves. In leaves, it is reduced to form the ammonia that finally form the amine group of the amino acids. The nitrate present in the soil is also reduced to the nitrogen by the process of the denitrification. Denitrification is carried by the bacteria, that is the Pseudomonas and the Thiobacillus. Now the 12.6.2 is the biological nitrogen fixation. Very few living organisms can utilize the nitrogen in the form of the nitrogen gas available abundantly in the air. Only certain prokaryotic species are capable of the fixing the nitrogen. Reduction of the nitrogen to the ammonia by the living organism is called as a biological nitrogen fixation. The enzyme is a nitrogenase which is capable of the nitrogen reduction and is present exclusively in the prokaryote, such as the microbes called as a nitrogen fixer. The nitrogen fixing microbes could be free living or a symbiotic. For example, the free living nitrogen fixing aerobic microbes are the Azotobacter and the Bergenkia, while the Rhodospirillum is anaerobic and the free living. In addition, a number of the cyanobacteria such as the Anabina, Nostoc are also free living nitrogen fixer. Now the symbiotic biological nitrogen fixation. Several types of the symbiotic biological nitrogen fixing associations are known. The most prominent among them is the legume bacteria relationship. The species of the rod-shaped rhizobium has such a relationship with the roots of the several legumes such as the alfalfa, sweet clover, then the sweet pea, lentils, garden pea, then the broad beans, clover beans, etc. The most common association of the roots is as a nodule. These nodules are the small outgrowth of the roots, and the microbes that like the Frankia also produces the nitrogen fixing nodules on the roots of the non-leguminous plant, for example the ulnus. Both rhizobium and the Frankia are the free living in the soil, but the symbionts can fix the nitrogen, atmospheric nitrogen. Uproot any one plant is the common pulses. Just before the flowering, you will see near the spherical outgrowth on the roots. These are the nodules. If you cut through them, you will notice the central portion is a red or a pink. What makes the nodule pink, and what is due to the presence of the leguminous hemoglobin or the leg hemoglobin? Now the nodule formation. Nodule formation involves the sequence of the multiple interaction between the rhizobium and the root of the host plant. The principal stages in the nodule formation are summarized as follows. The rhizobia multiply and colonize the surrounding of the root and get attached to the epidermal and the root hair cell. The root hair curl and the bacteria invades the root hair. An infection thread is produced carrying the bacteria into the cortex of the root, where they initiate and the nodule formation in the cortex of the root. Then the bacteria and release from the thread into the cell, which leads to the differentiation of the specialized nitrogen fixing cell. The nodule thus establish a direct vascular connection with the host for exchange of the nutrient. These events are depicted in figure. The nodule contains all the necessary biochemical components such as the enzyme nitrogenase and the leg hemoglobin. The enzyme nitrogenase is the molybdenum ferrous protein and catalyzes the conversion of the atmospheric nitrogen to the ammonia, the first stable product of the nitrogen fixation. The reaction is the N2 plus 8 electron plus 8 hydrogen plus 16 ATP. It gives rise to the 2NH3 plus H2 plus 16 ADP plus 16 inorganic phosphate. The enzyme nitrogenase is highly sensitive to the molecular oxygen and it requires the anaerobic condition. The nodule have adapted to that ensure that the enzyme is protected from the oxygen. To protect these enzymes, the nodule contains the oxygen scavenger called as a leg hemoglobin. It is interesting to note these microbes lie in aerobes under the free living conditions where the nitrogenase is not optional. But during the nitrogen fixing event, they becomes the anaerobic, thus the protecting the nitrogenase enzyme. You must have noticed in the above reaction, the ammonia synthesized by the nitrogenase required a very high input of the energy, that is the 8 ATP for each ammonia produced. The energy required thus is obtained from the respiration of the host cell. The fate of the ammonia. At physiological pH, the ammonia is protonated to form the ammonia, that is NH4 ammonium ion. While most of the plant can assimilate the nitrate as well as the ammonium ion. Latter is quite toxic to the plant and hence cannot accumulate in them. Let us now see how the ammonium ion is used to synthesize the amino acid in the plant, and there are the two main ways in which these can take place. Now first is a reductive amination, that is in this process the ammonia reacts with the alpha ketoglutarate acid and forms the glutamic acid as indicated in the equation given below, that is the alpha ketoglutarate acid plus ammonium ion plus NADPH in presence of glutamate dehydrogenase. It gives rise to the glutamate plus H2O plus NADP. Second is the transamination. It involves the transfer of the amino group from the one amino acid to the keto group of the keto acid. Glutamic acid is the main amino acid from which the transfer of the NH2. The amino group which take place and the other amino acid are formed through the transamination. The enzyme, that is the transaminase, catalyze all these reactions. For example, the ammonia act as a donor and plus ammonia act as a acceptor. It get exchanges and it forms the aspartic acid and glutamic acid. The two most important amides, that is the asparagine and the glutamine, found in the plant are the structural part of the protein. They are formed from the two amino acids, namely aspartic acid and the glutamic acid respectively, by addition of the another amino groups of each. The hydroxyl parts of the acid is replaced by the another NH2 radicals, since amide contains the more nitrogen than the amino acids. These are transported to other parts of the plant via the xylem vessel. In addition, along with the transpiration stream, the nodules of some plants, for example the soybean, they export the fixed nitrogen as a ureids, and this compound also have a particularly high nitrogen to the carbon ratio. Now the summary of the mineral nutrition. Plant obtain their inorganic nutrients from the air, water and the soil. The plant absorb a wide variety of the mineral element. Not all mineral elements that they absorb are required by the plant. Of the more than the 105 elements discovered so far, less than the 21 are essential and beneficial for the normal plant growth and the development. The elements required in a large quantity are called as a macronutrient, while those required in a less quantity or in a trace are termed as a micronutrient. These elements are either the essential constituents of the protein, carbohydrate, fats, nucleic acid, etc., and or take part in the various metabolic processes. Deficiency of each of these essential elements may lead to the symptoms called as a deficiency symptoms. The chlorosis, necrosis, stunted growth, impaired cell division, etc. Some are the prominent deficiency symptoms. The plant absorbs the mineral through the roots by their passive or the active process. They are carried to all parts of the organism through the xylem along with the water transport. Nitrogen is very essential for the substances of the life. The plant cannot use the atmospheric nitrogen directly, but some of the plant is associated with the nitrogen fixing bacteria, especially the root of the legumes, can fix this atmospheric nitrogen into the biologically unusable forms. The nitrogen fixation requires a strong reducing agent and the energy in the form of ATP. Nitrogen fixation is accomplished with the help of the nitrogen fixing microbes, mainly the rhizobium. The enzyme nitrogenase, which plays an important role in the biological nitrogen fixation, is very sensitive to the oxygen. Most of the processes take place in an anaerobic environment. The energy, the ATP required as provided by the respiration of the host cell. The ammonia produced following the nitrogen fixation is incorporated into the amino acid as a amino group. Thank you so much.